Hello, everyone. Well, my name is Asya Rose. I'm welcoming you here on Global IMMUNOTALKs. And today it's my special pleasure to introduce David Sancho. David? Hello, David.
Hello, how are you?
So good evening to everyone. Good morning, good afternoon. I'm really happy for this opportunity to present David. And David, his native country is Spain. His lab is in Spain, in Madrid. And he's there a professor in the National Center for Cardiovascular. And his clinical work specializes in immunology. And his research is kind of going through several levels. In his postdoc that he did in London, he found a certain immune C-type lectin that actually marks the dendritic cell subset, and it is involved in cross-presentation of antigen in MHC class 1. But then he moved back to Spain, to Madrid in 2010, and started his lab with the generous grant of ERC starting grant. And we know that in Europe, it's a very prestigious award recognition. And this starting grant was to investigate sensing of tissue damage by immune cells. He then continued, they got also the ERC Consolidator grant. And his research basically focuses on understanding how mitochondrial metabolism regulates dendritic cells and macrophages function, I guess with the long-term goal of improving immunotherapy. And he also explores molecular pathways involved in trained immunity in macrophages and the connection to atherosclerosis and the role of gut microbiota, which I know in the progression of atherosclerosis. So without further ado, I think this is kind of introducing your CV, but to get to know you a bit more personally, we would like to ask you, what is your mentorship style? How do you describe that?
Thank you, Asia. Thank you for the introduction, and also thank you to organizers for the nice invitation. It's really a pleasure to be here and to talk to you a little bit about our work in the last years, about these topics that you have mentioned. And yeah, about the mentorship style, maybe it's a question that you would better ask to the people in the lab, because I mean, one thing is what we try and the other thing what we achieve. But anyway, what I try as mentorship style is, first of all, to adapt to each one personality, to the unique personalities and needs of each individual in the lab, to try to foster their personal and professional development. But at the same time, I try to maintain objectivity and equitable dedication to all projects, because I think that it's very easy sometimes when you have a successful project, like you enjoy more spending most of the time in that successful project. And I think that is also very important that actually we dedicate most of our time to those projects that are not so successful, that need troubleshooting, and that actually need also further motivation for the people that are involved in those projects. So it's a hard part of our work, but it's actually our work. So in a way, I try to prioritize dedicating this time to troubleshooting, and also try to have scheduled meetings with people so that really the opportunity to meet all the people is scheduled and is not ad hoc, depending on the success, how successful is the project. Then the other aspect that for me is very important in mentorship is to try to create an inclusive and participative environment where every voice is valued. And something that I tell people when they come in the lab is, as undergraduate, as PhD students, whatever, is that there are really no hierarchies in the lab, and everyone's voice is valued equally. We have all the same brain, and we can have the better idea can come from the best idea can come from the latest PhD or undergraduate students. So it's very important really that regardless of seniority or experience, everyone's contributions are valued and respected equally. And that way we have a kind of team feeling, and we inspire each other by working as a team. I think that that's the nature of science in a way. So I try to empower people to challenge ideas, to ask questions, to take initiative of their own projects that they are leading, and that they really feel that they are leading the project and not the leader and just a kind of, coordinator trying to give feedback to everyone, trying to boost everyone's projects, but they are the leaders of their own projects. And the other aspect finally is also to try to spend time out of work, also to try to foster informal gatherings, so that we have a relationship that goes beyond the work relationship, and that creates a sense of trust on each other. And I think that is helpful also to spark this kind of collaborative environment, because I think that this is a field that is very challenging, I say unpredictable, and we have to cultivate resilience and maintain a positive outlook, and all that helps having like a sense of community. And at the end, I try to serve as a bridge, basically, for each member, not only to learn and excel scientifically, but also to grow as individuals. A bridge for the next stage, and someone that contributes to their success in the long term. That for me is the best satisfaction that you can get out of being a mentor, to see how people that are from with you progress and are successful. Much more than success with publications or projects or whatever.
Yeah, I guess we kind of, every time we build our mentorship style from looking at others, and thank you very much for sharing that, because it's a kind of putting the perspective for also for those who are trying to build their own. So thank you very much, David. I will just open the stage for you. Please feel free to share the screen. Just to remind you, in the end, there are no questions. There's going to be a bit abrupt ending. So, but tune with us. We are going to direct you to how to ask the questions. Sorry. Yeah, David, you want to ask something?
Yeah,
you're good?
Perfect. Yeah, I'm ready to go. Are you seeing my screen and my
Yes, we can see the screen. Yes.
Perfect.
Thank you.
So let's go ahead. Thank you so much again, Asia, for the introduction and the opportunity. And basically, what I want to discuss with you today is about our latest results in the last year, particularly since I established my lab already like a few years ago. And then I put this kind of general title to try to cover everything, but in fact, it's not very successful. But anyway, it's about exploring sensing on one hand and how this sensing affects metabolic adaptations. And because these are the main topics in which I want to focus. To start with this, I mean, basically, I want to clarify what are the cell types that are my preferred cell types, that are those that are immune cell types that are responsible for the initiation and control of immunity and inflammation, that are dendritic cells and macrophages, these beautiful cells. And then it all started actually many years ago, like 35 years ago. Charles Janeway proposed in this relevant publication that the pattern recognition theory, so basically pathogen molecular signatures are sensed by pathogen recognition receptors expressed in many cells, including myeloid cells, including dendritic cells and macrophages. And that would lead, in the case of myeloid cell, to myeloid cell activation, and therefore to the generation of a kind of effector myeloid cell that, in the case of dendritic cells, can promote activation of T cells and therefore generation of a T response. And in the case of macrophages, can contribute also to inflammation. However, we know that inflammation and immunity can occur in the absence or apparent absence of pathogens. And this happens in the case of anti-inflammatory or autoimmune diseases. So then there was at the time a very nice discussion, also fostered by Polly Matzinger, proposing that rather than just pumps from pathogens or microbia, it was important to consider that there could be also danger signals within our healthy tissue, danger signals that are performed endogenous adjuvants that are sequestered inside healthy cells, but eventually can be exposed or released upon necrotic cell death. So the idea now is that there could be forms of death even in our organisms, for instance, necrosis that could be immunogenic and therefore could lead to dendritic cell activation or macrophage activation, contributing to the inflammation or the adaptive response. So this is a nice proposal, but there was a lack at that time of receptors that could be involved in this pathway, and also to know what could be the effector responses to these danger signals and the relevance in vivo. So we contributed a little bit to this field by finding a receptor, as you have introduced. When I was doing my postdoctoral stay in Caetano de Sousa lab at the London Research Institute, we found a receptor, DNR1, also known as Clegnan-A, We call it actually DNGR1 because we thought that it could behave as a kind of danger receptor that was involved in cross-presentation. And basically, I just want to highlight like kind of the key experiment and try to make the presentation very simple so that also people from different fields can get the flavor of our work. So basically, this kind of cross-presentation assay is used as 3 cell types, basically, or three components. First of all, this is the dendritic cell that is a free ligand CDC1 that is basically a subset of dendritic cells that expresses this CLEC9A. And that is particularly good at cross-presentation. Then you have the T cells, in this case OVA-specific T cells, or T1, that are responding to ovalbumin, that you know that is the favorite antigen of the immunologist. And then you have these OVA GFP expressing UV dead cells. So basically, these are mouse embryonic fibroblasts that are expressing OVA, but cannot directly present OVA because they have this haplotag H2KBN1 that does not has a mutation that prevents the direct presentation of OVA. So basically, with this system, only OVA from these cells can be cross-presented through dendritic cells to T cells. And then we are going to see the response of T cells through the CFLC dilution, the T cell proliferation. So as a control, we use cells that do not express the antigen, and basically you don't have any proliferation of T cells. Then by using these T cells, these MEFs, expressing the OVA antigen, and that were UV dead, so they were necrotic, and they were secondary necrotic to apoptosis, then we saw that there was a very nice CFLC dilution, very nice cross-presentation, very nice proliferation of these T cells, because the antigen is cross-presented in CDC1s, but however, this was totally lost in the NGR1 deficient mice. In this case, the NGR1 deficient dendritic cells. However, when we were going, we were giving the antigen in OVA-coated beads, this OVA antigen was nicely cross-presented equally. So only when the antigen was associated to dead cells, there was an effective cross-presentation. So then we went further to demonstrate that this was happening in vivo. And indeed, we injected this OVA GFP UV radiated, so these kind of necrotic cells, to mice, and then we could induce tetramers that are CD8 cells responsive to this ovalbumin, and also reveal interferon gamma production of CD8 T cells in response to ovalbumin. And this was partially prevented in the absence of the NGR1. So basically, part of the cross-presentation in vivo is mediated through CLEC9A. So once we demonstrated this, of course, I started my independent lab, and one of our first questions, we were very interested in how dendritic cells could be sensing this tissue damage through the NGR1, and particularly in the context of pathogens and microbiota. So basically, one of the main questions that started the lab in 2010 was how dendritic cells and macrophages would sense tissue damage, particularly in the context also of microorganisms, both pathogenic microorganisms, but also microbiota. But then in a few years, we also became interested in how these innate signals, how this sensing of tissue damage and pathogens could lead to the modulation of metabolism in macrophages and dendritic cells, and how actually we could also manipulate mitochondrial metabolism in particular to influence dendritic cell and macrophage function. And at the end, our objective is always to improve immunotherapy, so to find new tools for immunotherapy. These are the people actually that are now in the lab working in all these areas, but there were many people through these years, and they all had particularly PhD and postdocs have first author publications, but I don't have time to go through all their publications and all their fantastic work, but I will focus just in a few works so that you can have a flavor of what we have done. So basically, one of the first questions that we had in the lab was, okay, we have detected a receptor, we have found a receptor, the NGR1, that detects this kind of immunogenic cell death, necrotic cell death. But then what happened in the context of pathogens? So if there's a pathogen that is inducing this immunogenic cell death, is the NGR1 still promoting just cross-presentation, or is also affecting activation of the dendritic cells? So this was a question that was unsolved. And then we went through the system led by Salva Iborra in the lab, a very talented postdoctoral that is now scientific director as a biotech company. And then he basically generated a line, raw vaccinia UV raw line, you know it, is a macrophage cell line that is from valve C. So if you have an antigen that is in this cell line, it's not going to be presented to B6 because there's a difference in the haplotype. So you can infect with vaccinia, and then if vaccinia, in this case vaccinia virus, is alive, then you're going to have direct presentation. But if you UV irradiate these infected cells, that inactivates the virus, and now the only way this antigen associated to vaccinia can be presented is through cross-presentation. And all this is detected by dendritic cells that have the NJR1 receptor that signals via Seq. So now our first question was, okay, is this treatment with raw vaccinia UV treated, so the virus is inactivated, or raw vaccinia, so the virus is alive, or just poly-IC as an adjuvant, is affecting activation of the dendritic cells depending on the NJR1 or the Seq pathway? And the answer, as you can see here, is that there's no difference. So that basically the activation of dendritic cells induced by the virus or by an adjuvant as a control is independent of the NJR1 or Seq. So that in this kind of a scheme, the NJR1 is not really participating in dendritic cell activation, because the important thing for dendritic cell activation are the pumps from the virus. However, is affecting then cross-presentation in this context of having a pathogen. So to do this experiment, we generated, again, this system with 3 components that are the meal, let's say that the cells that are infected, so are raw UV irradiated without antigen, raw infected with vaccinia, or raw infected with vaccinia and UV irradiated. So the only way the antigen can be presented to dendritic cells is through cross-presentation. So in this case, it's direct presentation. In this case, it's cross-presentation. And then we use dendritic cells that were wild type or the NJR1 deficient or deficient in the Seq pathway in dendritic cells. And then as T cells, we use T cells that are responsive to vaccinia because they are coming from effectors of mice that have been immunized with vaccinia. So then the readout here is to test how these T cells in response to antigen presentation are producing interferon gamma. And you can see nicely here that when there's no antigen, there's no interferon gamma production. When there's antigen coming from vaccinia and direct presentation, there's a nice production of interferon gamma in up to 20% of the cells, and there's no difference in the absence of the NJR1 or the Seq pathway. However, when the system was dependent on cross-presentation, the production of interferon gamma is lower in the control, but particularly is abolished or nearly abolished in the absence of the NJR1 or Seq. So what this is telling you, and by the way, this also occurs in vivo. So if you transfer this raw vaccinia UV in vivo, and then you measure interferon gamma production in response to restimulation with B8R or A3L that are peptides that are immunodominant for the response to vaccinia, in this context of cross-presentation, because these are cells that are infected but are irradiated with UV, so you don't have live virus. The only way to present the virus is through cross-presentation. Then you detect that there's really a deficiency in interferon gamma production in the absence of the NJR1, in the absence of Clegran A, and therefore that the NJR1 is also crucial for cross-presentation of vaccinia antigens in vivo. So what this is telling us is that even in the context of infection, you can, let's say, separate or segregate what is the dendritic cell activation that is dependent on the pumps of the virus from, and that is signal, let's say signal 2 or signal 3, production of cytokines or costimulatory molecules, from the signal 1 that is antigen presentation, antigen processing, and cross-presentation that was dependent on the NJR1. And therefore, this illustrates that the NJR1 and in particular in tissue damage sensing could contribute to this kind of pathway of antigen processing, and maybe in vaccines also would be important to foster this kind of danger signals to stimulate this antigen processing that is also a very important signal for induction of immunity. Also, these results that we published in this article were confirmed by the group of Caetano in a different viral infection, HSV1, and also they also found that this kind of dependency on the signaling motif for activation, so that DNGR1 is not really mediating activation, which is different from a very close receptor that is the actin-1. Then the next question that we were interested on this regard is that vaccinia can infect dendritic cells directly, or indirectly can infect cells that then are phagocytized and then leads to cross-presentation. We just have seen the examples. So, but the point is that this direct presentation is much more efficient than the cross-presentation. So why would be needed cross-presentation? And particularly, what is the role of cross-presentation and CEC1s in the immune response against vaccine? So to make the story very short, because I don't really have time to explain in detail every contribution, but basically what we found here, and I just described the final results, is that when you have this kind of infected and dead cells with vaccinia, they are processed mainly through CEC1s, through cross-priming of CEC1s, whereas direct priming is mainly mediated through CEC2s. So when you analyze the effect of these, effector T cells actually are generated by both CEC1s and CEC2s. And the same occurs with circulating memory. But resident memory precursors were particularly generated through this cross-priming through CEC1s. So basically, we had a deficiency in these CEC1s or cross-priming mediated through DNGR1. We had a partial deficiency in generation of resident memory cell precursors. So what are resident memory? Resident memory, basically, the typical memory that we have circulating in the lymphoid organs are circulating memory. Resident memory is the one that we have in the tissues, and that is particularly good at providing superior immunity to infection. Upon reinfection, if you find in the tissue already cells that are resident memory and are responding, they can really orchestrate a superior response to reinfection. And it's actually what we would like to have in to obtain better vaccination. So it's really important, therefore, to foster cross-priming, to foster this resident memory and improve vaccination. But also, not only to infection, we were also interested whether this could be important in the context of tumor immunity. And then in this regard, with the talent of Michel Enamorado, that is now an independent group leader at Mount Sinai, Michel basically, he found that TRM could contribute to tumor immunity. And this was one of the first papers suggesting this concept that now has been validated in many systems. And I just want to illustrate this experiment because I think that it was one of the most illustrative experiments to demonstrate this. So you have this model in which if you inoculate vaccinia, in this case, vaccinia ovalbumin, so that we can track the response to ovalbumin. You inject vaccinia ovalbumin by skin scarification or intradermally, you generate resident memory, but you also generate circulating memory. So now the idea is to take a naive mouse to do parabiosis so that they can share the circulating memory, but not the resident memory that will be only in these mice. Now, once they have been together for 30 days, we can split them, let them recover for further 30 days, and then inject B16 ova tumors intradermal. And because we have vaccinated here with vaccinia ova, it's going to respond against ova, and therefore it will control the B16 ova growth. So now, first control is to check that circulating memory indeed is the same after parabiosis, and indeed we found the same level of circulating memory. And however, as is defined, resident memory would be only because it's sessile, it's not transferable from one mouse to the other. Is only in those mice that were originally vaccinated. So now the experimental question is how the tumor develops. And now we have the B16 ova, we inject the B16 ova in both groups of mice. And then here you have the development in naive mice that basically haven't seen the tumor, the vaccinia ova, and therefore they develop the B16 ova quickly. But then any of the mice that has both mice, those that have resident memory and circulating memory, And those that have just circulating memory, these are the ones that have just circulating memory, here in red. And these are the ones that have also resident memory. As you can see here, both present immunity. So TCM alone, circulating memory alone, can promote really, indeed, anti-tumor response. But now, if you have also circulating memory plus resident memory, you see that the tumor taking is delayed, basically. So the tumor taking is delayed, and there's an improvement in anti-tumor immunity. So basically, what we demonstrated in this article is that both circulating and resident memory are sufficient for tumor rejection, but that the resident memory really can collaborate with circulating memory for an improved anti-tumor immunity. Actually, in the tumor, you can find cells that are, upon transfer of circulating memory, they can generate resident memory in both upon viral infection or in tumor challenge. And also we demonstrated that CEC1s were required for TCM for the activation of circulating memory against tumors. So we became really interested on the role of CEC1s in tumor immunotherapy. And for this, we collaborated with Nacho Melero, that is a very established researcher in the area of cancer immunotherapy. And then basically what we did, and I try to illustrate with this experiment, is the role of CEC1s in the checkpoint antibody therapy. So basically, we injected here MC38 intradermally, and then we injected antibodies that are like anti-CD137, that is an agonist antibody, or anti-PD-1, that is a blocking antibody. So both promote the release or the enhancement or the release of the brakes. And then they both collaborate. And actually, as you can see here in the growth of MC38, upon checkpoint antibody therapy, you can see that anti-CD137, anti-PD-1, and particularly the combination of both, are very effective to prevent tumor growth. But now we did this same experiment in mice that we are lacking CEC1s, that are BAF3 deficient mice. And then what we found was that then the antibodies were not effective anymore in preventing the rejection of the tumors. So basically, these BAF3 dependent CEC1s are crucial for basal anti-tumor immune response that will be then rescued by this kind of immunomodulatory antibodies. And then this led us to conclude that maybe if we boost CEC1s, we can have the opposite effect. We can improve tumor immunotherapy. So what we did here was to inject mice with free ligand plasmid, that is basically a therapy, gene therapy with a free ligand plasmid, so that free ligand is induced, and this free ligand is the cytokine that is expanding disease, and particularly CEC1s. And we injected the B16 nova, and now we treated with anti-PD-1 also with poly-IC, because poly-IC is an adjuvant that is particularly targeting TLR3 in CEC1s. And we tested the effect in tumor growth. And as you can see here, compare with just anti-PD-1, if you do the treatment with free ligand plus poly-IC, or even free ligand plus poly-IC plus anti-PD-1, you have a delay in tumor growth and even some survival in some of the mice. So with this, we conclude that if we expand and we activate CEC1s, this can synergize with anti-PD-1 in delaying tumor growth. And this can be really very important for checkpoint antibody therapy. But then to take this, the idea is to take this to humans and to improve, therefore, anti-cancer vaccines using CEC1s. We have to consider in the story of, I mean, there has been a long story of DC vaccines, anti-cancer DC vaccines. But all this started with monocyte-derived immature or semi-mature dendritic cells and progressed to other type of maturation with maturation cocktails using different kind of antigens. But this really has not been so effective as we would like. So we really need to go into the next generation DC vaccines. And now the tendency that we want to support with our results is that we should use rather natural disease and combination of different subsets, specific subsets of disease that are maybe not particularly this monocyte-derived disease. And also what is important is how to load these dendritic cells, particularly with which kind of antigens we should use. Now is important to identify neoantigens and also load with neoantigens peptides. But also in the absence of these peptides, we can be agnostic and take then kind of tumor cell lysates that are containing all these tumor antigens as a proof of concept at least. And then also it's important to generate this kind of immunogenic cell death that we know that is also important to provide other signals for antigen presentation and cross-presentation as I have defined. So to do this kind of assays and explore this area more in the preclinical arena, Steffi started this project, this collaboration in the context of a consortium also with clinicians to take this to the clinic. And Steffi, that is also a super talented postdoc that is now a group leader at Institute for Research in Biomedicine in Barcelona. And then basically what she tried was to optimize a vaccine based on tumor antigen-loaded CEC1s. For this, there are three components. First, the dendritic cell that we chose CEC1s. Second, the adjuvant that you use to activate the dendritic cell, and we selected poly-IC as an adjuvant that was able to induce really more CD8 T cell activation and proliferation. And then the source of antigen. And we selected a source of antigen, this tumor cell lysate. And to prepare this tumor cell lysate, basically, that we also obtained a high CD8 T cell response, we basically took B16 ova tumor, and we irradiated and also lyse by freeze and thaw cycles. And with this, we loaded CEC1s, and then we activated the CEC1. So these are CDC1 vaccine, basically CEC1s from the spleen, loaded with tumor cell lysates from B16 ova, for example, and then activated with poly-IC. So using this CDC1 vaccine, now we can treat B16 ova tumors. So we inject the B16 ova tumor intradermally. We treat with 1 million CEC1s. 5 days later, when the tumor is already 25 millimeters square area. And then 3 days later, we do a tumor and tumor draining lymph node analysis. And then what we can find, first of all, is that we have CD8 T cells that are specific for ova in the tumor, inside the tumor. And then the other aspect that was important is that if we were taking tumors early at that time, we already saw that there was an effect in the growth of the tumors. And even this was, if you leave the tumors for longer, you can see that there's an effect in survival. So CEC1 was working in this context of CEC1 vaccine was working in this context of B16 ova. But we wanted also to go to another model where we could combine this with anti-PD-1 therapy. And then we went to the MC38 model. So we treated in this case with MC38 lysate. And we generate a CEC1 vaccine that contains MC38 lysate plus poly-IC as an adjuvant. Now we treat with CEC1s vaccine plus minus anti-PD-1. And here you have examples of what happened. Here you have a growth of the tumor in the control. Anti-PD-1 is effective in reducing. CEC1s are effective also in reducing the tumor size, but particularly they both synergize. And this is represented here in the tumor growth with the representative experiment. As you can see, both CEC1s and anti-PD-1 have a therapeutic effect, but the most important thing is that they synergize and they increase, therefore, the survival. So what this is telling us is that right now, let's say the gold standard therapy that we have is checkpoint antibody therapy. What we are putting here, saying here, is that CEC1 vaccine can potentiate checkpoint antibody therapy. So it's really important and crucial, and we really should develop DC vaccines because they can really take us to a different level of protection together with this checkpoint antibody therapy that we have ongoing, and they can increase this, let's say, survival plateau. That is really what we are looking for in the context of cancer. And then for the second part of the talk, I want to focus more on some stories connecting this mitochondrial metabolism with myeloid cell function and how also innate sensing connects with metabolism. That is another aspect that we have developed in the last years in the lab. So I don't want to go into detail into all the pathways, metabolic pathways, etc., that probably you have forgotten since you studied your biochemistry. But the simple idea here is to say, okay, something as simple as glucose is metabolized through a pathway that can lead to glycolysis, and then this glycolysis can generate a lactate by fermentation, or can generate pyruvate that then enters into the mitochondria, into the tricarboxylic acid cycle, and then ultimately donate electrons to this electron transport chain. So if you see this metabolism is like a flux, where it's a flux of electrons, where the ultimate acceptor is the electron transport chain. So this electron transport chain in the inner mitochondrial membrane is composed by many complexes, where complex 1 and 2 are the main, let's say, entrance of electrons coming from NADH or FADH. And then these electrons are transferred to complex 3, 4, and 5, and so, well, complex 3 and 4, and then eventually they are transferred to oxygen so that they generate water. That's why we respire. And then all this transfer generates a proton pump into the intermembrane space, and this gradient is released through the complex 5, the ATPase, so that you generate ATP. So this is the main function in life of mitochondria, that is the generation of ATP. But if you see this, you will realize that also the function of the mitochondria is to keep this running. So to keep running metabolism, to keep running the regeneration of ROS. So at the end of the day, metabolites and redox balance are also controlled by mitochondria. And through these metabolites and redox balance, mitochondria is not only controlling energy generation, but also controlling functional responses. So what this has become clear in the last 10, 15 years is that not only normoxia or hypoxia or nutrients are regulating metabolism, but also there are innate signals that can regulate metabolism. And just as an example coming from this review, helminths can regulate more the TCA cycle, oxidative phosphorylation, whereas, for instance, danger signals through pumps and pattern recognition receptors can promote hypoxia and then promote this glycolysis and pentose phosphate pathway, and therefore lead to a rapid ATP generation and biosynthesis. And these OXPHOS can be linked, for instance, to more M2 macrophages, homeostatic macrophages, where this kind of glycolytic response can be linked to more pro-inflammatory M1 macrophages. This kind of the paradigm that there's in the field, but as every paradigm is a very simplistic view, no? So what we contributed early at that time, many years ago, one good thing actually of being here at the Cardiovascular Research Institute is that there are not many groups doing immunology actually, but there are many groups doing other stuff, like for example, mitochondrial biology. So we became interested in mitochondrial biology and how not only nutrients, that was what the other lab was studying, how nutrients were influencing the reorganization of mitochondrial complexes, but we hypothesized that maybe also sensing pathogens could be affecting, or sensing microbia could be affecting the reorganization of mitochondrial complexes. So we tested with dead bacteria, and actually dead bacteria were not really affecting mitochondrial metabolism or mitochondrial supercomplexes. They were producing this kind of glycolytic switch that is well described and is promoting antibacterial responses. But then what we found is that live bacteria, through bacterial RNA, is activating the production of ROS, and this reactive oxygen species, particularly in this context, would be activating this FGR kinase. These are SAR kinase, and then by the time this mitochondrial biology lab, just by the corner in the institute, they were studying how FGR could actually, this SAR kinase, phosphorylate complex 2 in the inner mitochondrial membrane. And this phosphorylation of complex 2 will lead to increased activity of complex 2. So we tested whether we would have also increased activity of complex 2 in the response to live bacteria. And indeed, we found increased complex 2 activity, and this would lead also, through red probably, to complex 1, that is reverse electron transport, to complex 1 destabilization. So this increased complex 2 activity was also linked to increase and enhance antibacterial responses beyond the responses that you would get to dead bacteria. For instance, more IL-1 beta production. So this was actually confirmed also with similar models, a bit different model, more in LPS actually, but was confirmed that the activation of complex 2 was leading to this kind of IL-1 beta production. This was confirmed by Luke O'Neill in a separate paper. So we became also interested how FGR kinase and complex 2 activity could influence not only the response to microbes, but also macrophage activation. We continued our collaboration with Toño Enriquez and Rebecca. And then what we found, we generated bone marrow-derived macrophages from wild type or FGR deficient mice, and we tested how was the polarization, the classical polarization and one polarization of this wild type versus FGR knockout or in response to different inhibitors. As you can see first, if you have a typical polarization with LPS plus interferon gamma, you could have increased MHC class 2, increased INOS, or increased IL-1 beta production in wild-type bone marrow-derived macrophages, and this was prevented in the absence of FGR, as you can see here in the red dots. But then, if you were doing this in the presence of N-acetylcysteine, that is a general Ross scavenger, MitoQ, that is a mitochondrial Ross scavenger, or these inhibitors that are complex 2 inhibitors, you were preventing in the wild type this induction of this of N1 polarization. So basically, the ROS FGR mitochondrial complex 2 axis would be regulating this macrophage polarization. So we decided to test a model where macrophage polarization could be important, as is high-fat diet-induced obesity. And then we found that in the FGR deficient mice, you had less weight gain, and you have this less weight gain because you have less fat. As you can see here in the MRI images, that the white color indicates fat, and you have more fat accumulation in the wild type compared to the FGR deficient. So you have this reduction in fat, and you have this reduction in fat because you have less Crohn-like structures. These Crohn-like structures are macrophages that are pro-inflammatory macrophages surrounding dead or dying adipocytes. And you can find this in the fat, and also you can do liver oil redox staining, that is staining the lipids in the liver, and therefore you see here that you have also reduced steatosis in the liver. So basically, FGR deficient mice show improved lipid metabolism, both in the adipose tissue, but also in the liver. And therefore, the idea is that FGR deficient mice do not develop metabolic syndrome upon high-fat diet, and the livers would have increased fatty acidosis and reduced steatosis. And therefore, if you go with a high-fat diet, in the presence of FGR, you have more obesity, but not in the absence of FGR. Mechanistically, we found that it was because the increase in a wild-type situation, you have an increased ROS in the context of obesity that would activate FGR and activate complex 2, and this would lead to N1 polarization, and this was prevented in the FGR knockout deficient situation. So as a last story, I would like to share our most recent results on how mitochondrial metabolism, a bit more in detail, about how mitochondrial metabolism of macrophages changes depending on the organ. And this, again, was a story led by the super talented postdoc Steffi Chulek in the lab, that now is an independent researcher at IRB Barcelona. So the general idea, the initial idea, is that there's a diversity of tissue macrophages type and functions, and you find different macrophages depending on the tissue with a different function in homeostasis, but also in the context of disease. But now the idea is what makes macrophages different depending on the tissue. What are the main features that characterize a macrophage depending on the organ? And then to analyze this question, we went to publish data, actually on, these are very good resource, on at that single cell level is a human cell landscape. So you have every organ characterized at the single cell level, and therefore you can take the information of macrophages from every organ. And then we took actually the information from macrophages from every organ, and we filtered the tissues, the organs, based on the number of tissue macrophages, so that we only took organs that contain more than 400 macrophages. And now we build a pseudo-bulk sample, putting all the information together, and we did a PCA analysis, just seeing how these macrophages would segregate depending on their organ location. And indeed, what we found through this PCA analysis is a very nice the location of macrophages depending on the organ. Its color is a different organ. So now, what is in this PC analysis? What are the main features? And we were surprised when we found that actually it was OXPHOS genes, the main signature found both in PC1 and PC2. So basically, OXPHOS gene would tell you where is located a macrophage, in which organ is located a macrophage. This was used in the human cell landscape. So through this single-cell RNA-seq analysis and just analyzing the macrophage cluster. But then you could also test the IMGEN project. In this case, the information is different. Is RNA-seq, bulk RNA-seq of macrophage populations. And now you can take this RNA-seq from different organs and again compare different organs and see what are the main genes that show a difference. And in this case, again, OXPHOS genes were in the top of the difference of macrophages among different organs. So mitochondrial oxone metabolism is a distinctive signature of macrophages in different organs, both in human and mouse. And if we know how is the OXPHOS of a macrophage, we may predict which is the organ where it's located. Indeed, for instance, there were macrophages that were highly dependent on OXPHOS. As you can see here, this is just concentrating in those mitochondrial genes. So these are just mitochondrial genes. These are just genes that are encoded by the mitochondrial DNA. And as you can see here, there's clearly a gradation of macrophages that are highly expressing those, whereas others are not expressing these genes. So now this challenge, in a way, the view, as I said, the paradigmatic view that you have an M0 macrophage that can be pro-inflammatory and then highly dependent on glycolysis, or homeostatic and highly dependent on OXPHOS, because you have homeostatic macrophages, because all macrophages in organs are actually homeostatic, but you have macrophages that seem to be highly dependent on OXPHOS, where others are not dependent on OXPHOS. So to test this, what we did was actually to interfere with OXPHOS in macrophages. And for this, we use the transcription factor for mitochondrial DNA that is important as a transcription factor, but also for replication of DNA. So we also mated this with drivers that are specific for macrophages, C11C3 and lysozyme-3. And then the TFAM is therefore affecting all the mitochondrial DNA and affecting all the mitochondrial proteins, encoded proteins that are affecting complex 1, 3, 4, and 5. So now, first thing is to validate the system. So depending on the driver, indeed, we found that we have a depletion of TFAM in macrophages, but now also validate functionally that this is affecting the DNA, the mitochondrial DNA levels. And indeed, it was affecting mitochondrial DNA levels, meaning that the TFAM is not acting as it should. So the system is working. Now, the first thing that you can do with this is just to take a look at the macrophages, to take a look at the numbers of the macrophages. And then we found that those organs where there's normally low expression of OXPHOS were not really affected by the interference with OXPHOS. However, those organs that are dependent on OXPHOS, were really affected in the numbers of macrophages contained once you interfere with OXPHOS in those macrophages. Particularly the winners, as you can see here, are the alveolar macrophages that, as we saw before, they are highly dependent on OXPHOS. So we did many of these experiments with alveolar macrophages, but also with other macrophages that are also dependent, like peritoneal macrophages or epidermal Langerhans cells. But I'm going to show just for the time the results in alveolar macrophages. So first thing that we did is, because you are depleting TFAM, TFAM is affecting mitochondrial DNA. As I said, mitochondrial DNA can be a danger signal. So we also try to validate our results by targeting individually each one of the complexes of the inner mitochondrial membrane, not just all of them with TFAM, but just targeting complex 1, complex 2, complex 3, or complex 4. And here you see the results. Complex 1 targeting was not really affecting the numbers of macrophages, but complex 2, complex 3, and complex 4 targeting was affecting the number of alveolar macrophages. So really the effect is mainly through this pathway, this complex 2, 3, 4. So what is the effect? Why TFAM deficient alveolar macrophages are actually dying and having an altered phenotype? So we found that actually they were not dying early at day 2 or 11, and they started to die after 3 weeks. So it's really not a problem in ontogeny, really. It's really a kind of cumulative toxicity that appears from 3 weeks onwards. And then in the adult, it's really evident. And together with this decreasing number, there was also a change in phenotype. If you were taking these 3 weeks alveolar macrophages that are still very similar in number, are however completely different in phenotype. So these are typical alveolar macrophage with high cyclic F and low C11b, and however this is totally immature in the absence of TFAM. So the maturity is affected, and so was in the when we were targeting individually each one of the complexes. So why is this happening? Well, then we went to 3 weeks, and this is important for all the students that are listening to this, that when we want to find a phenotype, we don't have to go to the situation in which we have already an evident phenotype, but really to the situation where the phenotype is starting. So we went to the 3 weeks where the phenotype was starting to test what was the difference at that time point. And then we found that we had reduced proliferation by Ki67 staining, but also increased apoptosis by caspase-3 staining. And why is they are dying or they are proliferating less? So we first thought that maybe there was a bioenergetic crisis, that if you are touching mitochondria, that's the first thing that you would think of. However, if you do this kind of respirometry assay, you see that while oxygen consumption rate is reduced, and particularly you see basal or maximal respiration rate here, they are reduced, but they are not completely gone. So it's not that the cells are not respiring at all. We are inhibiting respiration, but not completely. Then the other thing that you can see is that there's a switch to glycolysis. When you inhibit respiration, then there's a switch to lactate fermentation and lactate production. And therefore, at the end, if you measure ATP content, you have a similar ATP content. Only if you inhibit glycolysis with 2-deoxyglucose, you see a deficiency in ATP production. But in normal conditions, you don't have really deficiency in ATP. Therefore, the alveolar macrophages are not dying because they have a bioenergetic crisis. They are dying for by any other reason. So we started to hypothesize that maybe the reason was extrinsic, not really intrinsic to the macrophage, but extrinsic due to the environment where they are located. And that would explain why they are more sensitive or not, depending on the organ location. So we took alveolar macrophages from mice that were TFAM or C11C3 delta TFAM. So they don't have TFAM in the C11C3 compartment that is alveolar macrophages. And then we did the direct analysis at day 0, and we found again that there's less proliferation, that there's lower Ki67 staining, more ATF4 indicative of stress, less CDK1, less proliferation. However, now we can culture these macrophages for 48 hours. It's a very simple experiment just to release these alveolar macrophages from the environment. And then what we were very surprised to find is that actually many of these effects, like the Ki67, the proliferation, the stress, or the effect in cyclins, was reverted. So just by culturing these macrophages, just by putting them in culture, so just by taking them from the organ environment. So the source of cellular stress that leads to these impaired macrophages is extrinsic, is related to the organ environment. So what is the organ environment of a macrophage? Was basically the working life is to release, to phagocyte, to take this pulmonary surfactant. And therefore, we hypothesized that maybe this TFAM deficiency was affecting a cholesterol lipid handling activity of alveolar macrophages. And indeed, we found that in the absence of TFAM, we had more accumulation of lipids, as you can see here by the Nile red staining, or here in red. you have less oxidation of fatty acids, less efflux of cholesterol, and you have this kind of accumulation of cholesterol walls as you can see here, comparing the wild type versus the TFAN deficient alveolar macrophages. So the aspect is really different. And our conclusion is that TFAN deficiency would be leading to this OXPHOS dysfunction. And then this would lead to impaired fatty acid oxidation and cholesterol efflux. And then depending on the context where you have this OXPHOS dysfunction, if you have a context in which you have to deal with lipids, like for instance, to deal with this lung surfactant clearance, then this is going to lead to lipid and cholesterol accumulation, causing cellular stress and leading to cell cycle arrest and apoptosis. So basically, in homeostatic setting, OXPHOS is required for maintenance of macrophage populations with high lipid handling activity, depending on their environment. And the established paradigm, as I said, is that all homeostatic macrophages depend on OXPHOS. As you can see, depending on the organ, not all homeostatic macrophages do need OXPHOS. It depends very much on the organ environment and their lipid handling activity. So then we went to try to challenge, in a way, another idea, whether this could be targeted therapeutically and how would be this in the context of obesity. Because in the lean adipose tissue, we have seen that actually macrophages are quite quiescent and they are not really dependent on OXPHOS. But when you have high-fat diet, then you have the infiltration of monocytes that become this kind of pro-inflammatory monocytes, pro-inflammatory macrophages that are characterized by the expression of CD9 or C11C. And then when we did this kind of assay in which we took mice that are control littermates or those that are deficient in TFAN in lysozyme N, so deficient in TFAN in macrophages in the adipose tissue, and we submitted to high-fat diet for 9 weeks. Then we analyzed the macrophages in the EWAT, and here now, remember that in homeostatic conditions, we don't see differences in the adipose tissue in TFAN deficiency, but now we see deficiency upon high-fat diet. But this deficiency is not because of homeostatic macrophages in the fat, but because of inflammatory macrophages in the fat that are characterized by the expression of C11C and CD9. Also, you see a decreased number of Crohn-like structures that are formed by these pro-inflammatory macrophages. And also, if you test pro-inflammatory cytokines in the white adipose tissue, you can see that there's a decrease in the absence of TFAN. So basically, pro-inflammatory white adipose tissue macrophages are reduced in these TFAN-deficient high-fat diet-fed mice. This, of course, affects also to the weight gain, and you can see here that the body mass is reduced in the absence of TFAN, particularly because of reduced fat mass. And this also affects to the insulin profile. You can see here an insulin tolerant test, and if you have this lower curve, it means that it's a better profile. You can really lower glucose levels much more. And also in liver steatosis, you can see this oil redox staining, how it is reduced in the absence of TFAN. So basically, you have a situation in which the OXPHOS impairment in pro-inflammatory white epithelial tissue macrophages reduces obesity and prevents metabolic syndrome and hepatosteatosis because they are reduced in number. The established paradigm is that pro-inflammatory macrophages act on OXPHOS and rely on glycolysis. What we see here is that actually pro-inflammatory macrophages in obesity rely on active OXPHOS to deal with lipids, the excess of lipids. And if you interfere with OXPHOS, they die. And therefore, you have less pro-inflammatory macrophages and you have a leaner adipose tissue. So basically, in summary, we have seen that there's different tissue macrophages in homeostasis, and depending on the organ, they have a different dependency on oxidative phosphorylation. And those that are have more expression of oxidative phosphorylation are more vulnerable, as is logical, to OXPHOS loss, where those that have lower expression are more tolerant. So in this case, you can target homeostasis. For instance, in the absence of OXPHOS, you have alveolar proteinosis and you have impaired alveolar macrophages, but also in the absence of OXPHOS, you can target specifically pro-inflammatory macrophages in the adipose tissue, and therefore you can have leaner mice. This could lead to eventually some therapeutic approaches. So finally, just to finish, because this is a global IMMUNOTALK, also I want to give a flavor that we are working in Madrid. It's a very lively city, full of culture, and it's very sunny, but also can be snowy in the winter. And we do this work at CENIC, that is a cardiovascular research institute in Madrid. It's really a state-of-the-art institute with great facilities, and really indebted to these facilities, to all the collaborative environment that is in this institute, and also to the funding bodies, international and also national, particularly the Spain government, but also the CENIC and also the ERC at the European level, some consortium from the Horizon 2020, La Caixa Foundation, that is a great foundation, private foundation in Spain and Portugal for funding science, and also other banks and private companies and funding bodies. And finally, particularly, I want to thank to all the people that have made possible this throughout the years. Because as I said at the start, all of them PhD student postdocs that have gone through the lab have published as fair authors, but I didn't have really the time to go through all the detail of all their wonderful stories and wonderful work, and really indebted to their motivation to work and to do fantastic science. And thank you so much for your attention. Thanks to also all the dozens of collaborators across the globe that have made this possible. Thank you so much.
Thank you very much. It was really interesting, David. Thank you for sharing your work and sharing all the insights that you gather from the work. As you know, there are no questions in this format to allow everyone to communicate more directly with David, including with the time differences. So just for all of you, the way to contact David and to ask him questions is going to be via X, Twitter. And you need to search for an account which called Global IMMUNOTALKs, and then find a tweet that says, ask questions for David Sancho, and just contact him and he will be able to reply to your questions. And you can also find the direct handle to his lab. And I'm sure you're going to have a lot of questions because you raised a lot of very interesting directions that are so relevant for many of the researchers in this field. So with that, I would like to thank you all. Thank you, David, again. Thanks the organizer for staying, making it happen. And Fuzia, thank you. And I hope to see you soon, next week, in the next Global IMMUNOTALK. Bye, everyone.